There are several enzymes that modify the DNA molecules by addition or removal of specific chemical groups. Those are known as DNA modifying enzymes. Let's know about few DNA modifying enzymes one by one. First example of DNA modifying enzyme is alkaline phosphatase, which is obtained from E. coli or calf intestinal tissue. In gene cloning process, this enzyme is used to remove phosphate groups from the 5' end of DNA molecules. As you know, at the 5' end, phosphate group is present in DNA molecule, which is replaced by OH group at the 5' end of that DNA molecule by the action of alkaline phosphatase. This is also used as a reporter gene for identification of secretion signals. Second example is polynucleotide kinase, which is obtained from E. coli infected with T4 virus. Now, in this case, it has a reverse effect to alkaline phosphatase. That means it transfers phosphate of ATP to a 5' OH end in DNA or RNA. In this figure, you can see that 5' OH of DNA is converted to 5' phosphate group by the action of polynucleotide kinase, which is just showing the opposite action of alkaline phosphate. It is also used for the labeling of 5' termini of DNA for Maxam-Gilbert DNA sequencing method and phosphorylation of DNA lacking 5' phosphate termini. Third example of DNA modifying enzyme is terminal deoxynucleotide transferase, which is obtained from calf thymus tissue. It is a specialized DNA polymerase that does not require a DNA template in order to amplify DNA. You have seen that almost all of the DNA polymerases need template in order to amplify DNA. But the exception is in case of terminal deoxynucleotide transferase. It catalyzes the stepwise addition of single nucleotides or DNTP onto the 3' end of any oligonucleotide sequence. I have given two figures. One is for the single-stranded DNA, another one is for the double-stranded DNA, where you can see that this enzyme is catalyzing addition of DNTP at the 3' position of single-stranded DNA as well as double-stranded DNA. One of the primary uses of terminal transferase is the tailing of vectors and cDNA with complementary bases, thus permitting the cloning of the cDNA fragments. That means this is helping in the process of cDNA cloning. It can also be used for labeling of 3' ends of DNA fragments.